Okay. Hi, everyone. First of all, I'd like to thank the organizing committee of ISCB for the opportunity for presenting my work in this space. I am a master's student at Simvestab, and this is my project, Genomic Profiling of Bacteria for the Prediction of Synthetic Community Assembly under Antagonistic Interactions. And I would like to start with these three questions. Sorry. What is a community? What is community assembly? And why is it important? So let's imagine we have a collection of organisms, of individuals, and we know that these individuals interact with each other. And as a microbial enthusiast, I would like to use the example of microbes. That is a community, a collection. But there's also interaction happening, and this interaction leads to complex processes and complex dynamics. And that will determine who is there and how many are there. For the question, who is there, we have this like basic example, like 4 kinds of species. And how many are there are the abundances of them in this community, in this flask. Also, these communities may face environmental changes, temperature or nutrients, but also we have another complex variable, that is what happened if at some point in the time I add a newcomer, a new species. So then that gets into a more complex dynamics. This is really important nowadays with all of the microbiome research. The many applications of synthetic communities or the study of ecological processes in communities can be from human health. We have seen a lot of examples here. Also from bioremediation to the creation of or design of synthetic communities for agricultural crops or biodegradation of some residues. And now I'm going to talk especially to this community that we work with in the lab. So a former PhD student did a sampling in Churince, in Cuatro Ciénegas. Cuatro Ciénegas is an oligotrophic place. These waters are low in really so many nutrients, and their dynamics are really special. So what she did, she captured the Bacillota diversity of this place. And she isolated 78 strains of Bacillota. That's a lot of work. But that wasn't just enough. She took these 78 isolates and then performed an analysis of pairwise interactions. And these diagrams that I'm showing are like a representation of what does that mean to make a pairwise interaction of only 4 species. So that's the combinations that you have to do if you want to analyze only 4 species. So you can imagine what it's like to have 78. Also, she took information about this particular interaction, that is antagonism. And antagonism is defined as the ability to inhibit the development of other microorganisms in its community. So you can see that in the example of the bottom, the orange species is not allowing the purple one to grow. So you can imagine this matrix of 78 in presence or absence of this antagonism, but also some other people were saying, networks are everywhere. So we can visualize this matrix in an hierarchical network. And what we are seeing here, the first thing, each node represents a strain of the 78. Then we have another variable that is the degradation of the color from green to red, represents a metric that is called the degree of the network. So the degree, it means that if you are in the red spectrum, you antagonize, you are a really antagonized strain. And if you are in the green side, you are not antagonizing like many strains. So that's the representation of the color degradation, but also we have another thing that it's who's in the top and who's in the bottom. And the ones in the top are the ones that are antagonizing a lot of strains of the 78. And the ones in the lower like label are not antagonizing many. So what we are seeing here is that we have so many strains that are not attacking the other or antagonizing the others, and a little that are doing that. So I call it this in common words that the ones in the top are bullies, but they don't get bullied. So that's really interesting in the dynamics. Also, we have this broader process, but what happens if we take a small sample of these communities, take it to the lab, and build these synthetic microbial communities? So, a current PhD in the lab, Aguilar Salinas, he made so many combination of these 78, and he found a triad that have some interesting characteristics that I'll address here. So he studied this particular bacillus. Here we have pairwise interactions, Bacillus pumilus 145 and Subclifera horikoshi 28, in a specific proportion or ratio 1 to 10. So what we are seeing here are like death curves analysis. So is the CFU, that is the colony forming units. So he counts the units in the time, and we have 30 minutes. And the dotted line, the green dotted line that we are seeing is the green species, I'm gonna call it that way, the green species growing alone. So it remains constant, but if we have an interaction, a pair interaction with the orange one, we see that in the first like 5 minutes, it's dying. So that's what antagonism also is. We can see antagonism this way. But what happens if we add a third one? And in this case is Bacillus pumilus 111. And you can see again the dotted line that represents the green species growing perfectly fine. And then if we add the blue one, it's clear that we don't have this antagonism anymore. So something is happening there. And this model, because not all of the other combination were predictable, and this one was really special. So we call this the bars model. That is from Bacillota antagonist, resistant strain, and the sensitive one. So if I mention bars, I'm talking about this. And other important thing is that the Bacillus pumilus, the blue species, is in a very low proportion. It's in 0.5. So I put this only for you to really believe me that these other pair interaction are not affecting the dynamics. So it's really an antagonism and some kind of protection. So here I like to show you this core phylogeny of 1,000 core genes, and the SAR are pointing to the bars species. So we found in the 78 all of these phylogenetic groups. And again, we're seeing our network, our hierarchical network. And what we can see is that there are some groups, or I see clear groups from this antagonism network that is hierarchical, that the ones that are really antagonists belongs to a particular group. So now this is a heat map of biosynthetic gene cluster predictions. And what we are seeing here are the secondary metabolites and some bioactive important compounds and many toxin-like or antibiotic-like genes. So it's also a presence or absence of these categories. And we have in the left all of the species, and in the right, the phylogenetic groups, colored by phylogenetic groups. And in the top, we have the clusters that all of the classes made. So what we can see really clear is that they are forming specific groups of these biosynthetic gene clusters. So what does that show us? For example, I can't point, sorry, but in the top, we are seeing like a blue square, and they have only those biosynthetic gene clusters. So there are some things that are really unique of those species. And we are looking to antagonism. So for future perspectives, what we want to address is what are those differences. And in terms of these synthetic communities, what can we choose of this profile, of the genomic profile, of how can we explore more variables that the genomes contain in order to build from a more intuitive that we have information to build new communities that respect, for example, the bars. So what would we choose next if we have this profile and some other information? From these bars communities, we also have metabolomic and transcriptomics and other analyses. So I'm not going to address that here, but well, the key messages of this talk are that we found consistency between the distribution of clusters for bioactive compounds and the phylogenetic groups within these members of Bacillota. That's important because this is in this group. This coherence could be used as a start point for designing synthetic communities and manipulating population assemblages for practical purposes. However, more research is needed to understand emerging properties of these communities and to what extent it is possible to predict the results of the assemblage, as shown in the bars. And finally, I would like to thank my advisors. Also, all of these people are involved in this project, that the funding comes from CONACYT Fronteras de la Ciencia. So Lena Mexico was really key to have this whole genome sequencing. And also, importantly, I like to thank all of the women and all of the people that make these spaces that are free or safe for us to present and to make this interaction. Thank you very much. And I would like now to answer some of your questions.
Thank you, Marisol. So please, any questions? No? I have one.
Thank you.
Yes, I would like to ask you about, because in this, in your work, you consider binary interactions, and I understand this should be the first level of complexity. But what do you think about higher order interactions and how these higher order interactions may alter your results?
Wait, what, sorry?
Higher order interaction, instead of testing 2, but the interaction of 3 or 4, that is 3, 4, how this may alter the results that you are presenting.
Okay. So yeah, it's really, the intention of here is to predict some of the dynamics. And with pairs, we are seeing that it's not enough.
Yeah.
And with these profiles, we have some like clues maybe to get to understand these processes, but they are really mechanistic dependent. If this interaction happened because of a cell-to-cell contact or a diffusion of something else. So I think that we have to understand our small communities in order to try to build something like
to scale.
But also there are some models that these like the further steps of this project to build some models from cellular automaton that incorporates these other variables. So yeah, it's not as easy to predict from the genomics only or the experiments only. We have to incorporate a lot of things. Thank you.
I understand.